And that's what's gonna go to our exam 5. It's not a comprehensive exam, so it is exam for chapter 21, 22, and 26, okay? And then our final lab in the last week as well. So we only have 3 weeks left. So 6 lecture period, and of course only 5 really, because then there's exam. And then to remind you, this week is the last lab, physical lab that we're actually gonna do, and it's a wet lab. Because the following week we're doing the review.
Oh, wow.
Because the lab exam is comprehensive. But you do have your manual and 3 hours. So hopefully.
You can make something happen.
And the review, right? So that should be, usually people do really well in the lab exam. So if you're right there between the grade, lab exam is your savior. But, so I think I have entered everything I had except this point that I just entered. So your grade is how it is right now. You probably guys noticed the extra point assignments. Some people took it already and did it
And
already.
I don't know my grade because of my last exam.
Well, hey.
I'm the only one.
But yeah, no.
Oh, yeah,
There
there
are
two.
just 4 people.
Oh, 4.
So, yeah, any questions, guys, on how the semester's gonna go? The 3 weeks of it? I hope you're prioritizing your grade at this point, right? Throwing your energy into, no, I mean, I'm just because you're so, no, because you're so relatable, so I can just look at you. No, I don't mean anything by that. I just have to look somewhere. Anywhere I look, they're gonna say, why are you looking at me?
I know, I was like, did I miss something?
Okay, so shall we go?
Let's do it.
So for the Thursday, I will not have the question of the day. But on Tuesday, I have 2 questions for chapter 22, okay? But please spend energy because the metabolism is sometimes confusing to people. So I'm gonna let you off the hook on Thursday, but then on Tuesday, because you have more time, 2 questions of the day gonna be for the chapter 22, okay? You'll be warned, you were warned. All right, so digestive system, and again, series of the absorption, secretion, digestion, right? So more terms that some of you are familiar with, and some of them may mean different things in the digestive system. So as far as the mass balance, right? So most of what we put in the lumen, and who remembers what lumen is? Lumen, Israel?
Hell yeah.
Small hole. Can somebody do better? Brian? Huh? So most of what is put in the lumen is absorbed. So is it lumen? Yes, it is in blood vessels. But what else? I mean, can you do a more comprehensive?
Isn't it hollow space?
Hollow space.
Yeah, on a small.
It's the opening.
Yeah, give me examples. What would be a lumen in digestive system?
The small. No. Oh, the appendix. And then there's a little.
Why are you so confused, Vivian? So when you say esophagus, it's like an organ, right? But what would be the lumen part of it? The opening. The opening. The hollow part, right? Does the stomach has a lumen?
The stomach?
Yes. Does the small intestine has a lumen?
Yes.
Does the large intestine has a lumen?
Yes.
Does blood vessel has a lumen?
Yes.
Does bronchi have a lumen?
Yes.
Yes. Okay, lumen is that hollow part. So most of what is put into lumen is absorbed, meaning that it goes where? What do you think? Where does it go? Just think about circulation. I mean, digestive system right now. So what does that mean, absorbed?
It's in the microvilli of the small intestine,
And then what does that mean, absorbed? Where does it go?
in circulation.
In the circulation, right? So what we put in our mouths does go into circulation, right? So be careful about what you put in your
And the
mouth.
lymph as well.
Okay.
I mean,
So the rest would be excreted. If something is not absorbed, right, it will be excreted, right? So this is really not for our class, but if you're curious, there's a very cute poop guideline that you can check it out. Okay, so ingestion. The 2 liters of usually food and drinks, plus minus, of course. So this is meaning like we ingest it, right? Saliva throughout the day, 1.5 liter. Are you surprised? Does it seem like a lot?
No.
Like you guys picturing the liter bottle? So bile is a relatively small contribution. 2 liter of gastric juices, right? That's also a very large amount. All of that is actually into the lumen. All of that is put in the lumen, right? So pancreatic secretion with all the digestive enzymes from pancreas, that's a pretty big amount as well. And then intestinal secretion of various mucus and different compounds, so we'll talk about that later. So 9 liters, right? So then, if that's like an average, we put into the lumen, like the input into our system, about 9 liters, 7.5 is actually absorbed from the small and the 1.4 absorbed from the large intestine. So as you can see, majority is actually absorbed, and what's not absorbed is excreted. Very quick, most of you familiar with the basic anatomy. So oral cavity, that's where digestion begins for, as we know, for what? For carbs. And actually, your book kind of doesn't mention that, or maybe it was published later on, but it mentions, when we get to that chapter, when we get to that table that talks about what's absorbed, it kind of says there is no absorption, but there is a limited absorption in the mouth as well. I mean, otherwise our like under the tongue pills wouldn't really work, right? But definitely digestions of the carbs begin that. There is actually a small amount of the lipase. Again, book doesn't mention that because it's such a small amount. So I mean, technically, if the lipase is the breakdown of the fat, so they could start digestion here as well. But we mostly say carbs, and that's okay. On kind of general way of speaking to it. But I do want you to know there's a small amount of lipase and a small amount of digestion of the fats that starts as well. So the mastication is, of course, the chewing and breaking food into smaller pieces. That's what our teeth are for, as you know. Salivary gland and the production of the different compounds, but the one that's very important for this conversation is the amylase. So the amylase, that ending usually suggests that it's an enzyme. And not all enzymes have that same ending, but lactase, amylase, that is the enzyme that's gonna start breaking down starch. And we were just talking with Nardas about that, is that if you keep your cookie or bread in your mouth for a little longer, don't swallow too fast, it will become sweeter as it stays in your mouth because of the amylase breaking down the starch. And the glucose, which is the breakdown product of the starch, is sweeter than the starch. So the bread or the cookie will become sweeter as they being digested. So you can test it out sometime. And then finally, we call this processed food a bolus. And we call it bolus all the way to stomach. And after stomach, we call it something else. Anybody remember what's the name?
Chyme.
Chyme, yeah, or some people say chyme, yeah. All right. A few seconds in the esophagus and the stomach, and the food gets dropped to the stomach. We do definitely should remember that the protein digestion starts there. We have proteases, the enzyme that breaks down the proteins. So if the proteases are working in the stomach and the stomach pH is around 1 to 2, so then what do you think the optimal pH for these enzymes are? The protease is 1 to 2, right? Because like when you go into different digestive enzymes that's released by pancreatic juices, they actually prefer 7.3, 7.2. So it's interesting because in the stomach, the environment is very, very different, right? And as we know, the pH not only enables to kill some bacteria to a certain degree, but also assists digestion of the proteins. So, and denaturation of the proteins happen via acid. And remember I was mentioning to you, if you eat some of the seafoods, like especially like mussels and those kind of guys, bivalves, I guess the collective terms will be, squeezing the lemon juice, what kind of does the same thing, it's coagulation. So acid denatures the proteins, and the proteases will begin protein digestions. And at that point, that kind of soupy, smoothie-looking mixture is called chyme or chyme. The other organ that is very important, and accessory organ, that's not sort of like the part of the continuum, but it does release the pancreatic juices right into duodenum. And we'll talk about that a little bit more. It will have multiple enzymes and bicarbonate to neutralize the chyme, right? Because it comes in very acidic from the stomach. Those enzymes not gonna work. So we need to bring it back to the pH 7.372 for these enzymes to work. So the neutralizer also coming from the pancreas in the form of bicarbonate. Of course, liver via gallbladder will supply bile, and one of the key function there is that is emulsifier. And if you guys ever kind of encounter that word, you could have used it like when we used the dish soap, right? That breaks down the fats. We also that also kind of emulsification, because it breaks down big fat droplets into the very small one. And again, we'll talk about that in details. The overview here right now. So the bile is very important, and of course it carries some additional compounds that's going to be very important for fat metabolism. Most of the nutrients you guys notice as you were doing small intestines are absorbed there, right, in the small intestine. And very large surface area, of course, all this winding here, on average could be up to 25 feet. So it's pretty large surface area for all the absorption, and of course it is really to be able to absorb all the nutrients as they move along, right? Because the whole point of eating is to get all these nutrients that is necessary for maintaining the body. And in the large intestine, there is some absorption that's gonna take place as well. I'll mention that in a minute, but some nutrients absorption does take place, but it's mostly water electrolytes, some vitamin, and of course, a form of the stool. The other thing that's very important to remember, large intestine is the home for a lot of different bacteria, good and bad. We call it microbiome, and they do digest multiple foods that our body does not have a digestive enzymes for. And the products of the metabolism of the bacteria and the digestion byproducts is also absorbed into our system. So we do get absorption from the work the bacteria did in the large intestine. So hopefully it's very clear that absorption is one of the most important jobs, right? Getting all the nutrients out of the food. And we have a very special mucosa that is prepared to even further increase the surface area. There is a multiple rugae and plicae, like people say it very differently, but the pictures probably will help more. It's kind of this different types of folding. This is very, I'm sorry, this is kind of fuzzy. So the rugae, and then plica in the digestive, in the small intestine and large intestine. Let me get to the plica and then I'll go back to see this kind of folds. Because if you think about that, if you unfold that, right, it's going to be even larger surface area. It's all about increasing surface area. Because the larger the surface area, the more opportunities to absorb all the nutrients, right? And on top of that, look at this, there is yet an additional processes they call villi. So again, the rule is, the rule of the land is the greater the surface area, the better. So here's another view. This is the small intestine villi. So as you can see, there's very multiple area for absorption. And right under the mucosa, we will find different gut association immune system, the Peyer's patches. This is like a sort of the collection of the immune cells for surveillance. Of course, the nerves and the vessels, veins, and artery. We will have a multiple muscular tissues. We'll talk about that again when we get into a specific organs to promote the movement and the mixing of the bolus or chyme. So this is the macroscopical. So here's our submucosa. This is as you can kind of see the glands and the peats. And the rugae, as you can see, much larger folds of the mucosa. So here is our little villi, and then the entire fold is the plicae. Okay, so there are 4 key processes that happens in digestive system. So digestion itself, meaning breaking down the food into the manageable compounds so that we can actually absorb them, right? Because if you have a starch, which is a very large molecule, right? Multiple chains of glucose, it's not gonna be like crossing the barrier. It's not gonna enter, right? So if you have very large tertiary or quaternary structure of the protein, it has to get broken down to a smaller component, to a building blocks, so they can actually be absorbed. So digestion takes care of that. It breaks down the large macromolecules into the smaller building units, and we'll discuss to what are the minimum requirements for breaking down each micronutrient so that it actually can be absorbed, meaning come from the lumen into the circulation. And then secretion is actually the cells of GI producing something and contributing to lumen, right? That's in that direction, from the cells of GI into the lumen. And motility is the movement of the bolus and the chyme. So what are the challenges? How to digest the food and not digest the lining, right? Because the lining is obviously made of the different molecules as well. Balancing the mass balance, that you don't have the excess, like how much you put in, that so much gets used up. If the fluids are lost through the diarrhea, vomiting, what is the mechanism of homeostatically reversing that? And with food and drinks, opportunities for a lot of pathogens to come through, and so then how do we deal with that as well, right? So those are the main challenges and the work of the digestive system. So secretion, as we mentioned, movement of material from the cells into the lumen, or ACF. Now, when they say ACF, as you know, if it needs to go into circulation, it will. If it needs to stay in ACF, it could stay, but this is a pretty kind of open communication here because of the lining of the blood vessels are very, very leaky. So digestion, so mechanical chemical breakdown, absorption, movement of material from the lumen into ACF, and then of course into circulation, and the movement of material through the GI tract. So that's a good slide for you to remind yourself of all the 4 processes, then make sure you can recognize them. And so let's quickly mention motility because probably least complicated part, and then we'll get to others. So we distinguish different types of movement in the GI. The segmentation is really the work of the circular muscles, and if you look at the digestive tract, specifically maybe in the small intestine, it's easier when the circular muscles are really just the mixing the food by contracting like that. So it is really the mixing work. Peristalsis is really the movement forward, right? And the way the peristalsis works is that it's kind of very progressive movement where, again, if you have your, the small intestines here, the contraction will push the chyme towards, towards the end, and then, press again and push it forward. So it's sort of like this progressive movement that enables the direction, one way to go, right? So from one section to another. And this is a very interesting motor complex, migrating motor corpuscles. That is actually very interesting. It's sort of like a cleansing, a contraction, that series of contraction from stomach through colon. And that usually happens between the meals, so it's not activated by the chyme itself. It's more of the kind of sweeping, making sure there's nothing left in the digestive system, and because then it's subject to degradation. If it didn't get absorbed, then it shouldn't really move. So there's of course multiple problems that we're not going to be addressing here, and I'm sure you've heard some of them. The delayed gastric emptying is when it stays too long in the stomach. Let's say normally it should be 2 to 4 hours to leave the stomach, so that happens especially in the elderly patients. Esophageal spasm, constipation, diarrhea, irritable bowel syndrome. I'm sure you have some general awareness, but it's not really our subject for us to talk about here. So just kind of shows the peristalsis, the segmental contraction, and migrating motor complex. So this is like, I put this here for you a little bit to distinguish bolus and chyme. So the bolus from your, I just lost the name of the, from your mouth cavity to the stomach, oral cavity, just mouth, oral cavity. So it is more alkaline because of the saliva. It's quite alkaline. And then, so that's all the way to the stomach, and then bolus converted to chyme in the stomach, obviously more acidic because of hydrochloric acid, and it's more mushy, soupy, as opposed to the part that's a little bit, the bolus a bit more, a bigger pieces of the food, obviously. Interestingly, we do have this series of the neurons, a quite large number of neurons that work specifically within the enteric nervous system. And it's quite complicated, more complicated than we thought in the past. And it not only can get, of course, influences from medulla and the parasympathetic neuron, especially via vagus nerve. Yes, it does get a lot of communication with the central nervous system, but it can also work with the chemicals from the food and the chemicals released by the gastric and the intestinal mucosa, and it's actually capable of independent function, independent from the central nervous system. So enteric nervous system is actually quite popular right now for scientists to study and get a little bit more into it, because it wasn't recognized up until like maybe 10 years ago as much as it should have. So could actually work. We call it short reflexes. So short reflexes will be something that based on, let's say, there is extension of the stomach, distension of the stomach, or depending on what kind of food is ingested, the proteins, amino acids, fats. There could be series of short reflexes within the enteric nervous system to adjust the production of different chemicals and enzymes and cytokines, and deal with it locally without actually talking to the central nervous system. So quite interesting. This is a very, very good table for you guys to make sure you have that for your exam, because this is also most of the questions that you guys did on the question of the day came from here. And that's a really nice one because it kind of collects all of this together. So, and it keeps using these 4 main functions and kind of discusses the differences between different parts, right? So for example, in the oral cavity, right, saliva is the key secretion process. Carbohydrates, you could make a little note that minimal fat digestion because of the lipases. You could put it right here. And here where it says absorption none, if you can also please correct, say like, again, minimal absorption. Because again, you know, as you probably guys know, right? Putting anything under the tongue like nitroglycerin or the other drugs that are actually meant to dissolve very quickly requires the absorption into circulatory system. And motility would be mostly the chewing and of course deglutition, the swallowing effect. So other than that, the rest of it is pretty accurate. So here we are in the stomach, so secretion into the lumen. Hydrochloric acid, pepsinogen, which is the precursor of pepsin. And so pepsinogen is released, and then it requires the acid to actually turn it at pepsin, so then they can digest the proteins. Gastric lipases, that's where your fat digestion also begins, right? Mucose bicarbonate for protection. Mucus and bicarbonate is very important to protect the lining so it doesn't auto-digest. Many different other chemicals, we'll talk about them when we get there. So quite a numerous secretion over here. So mostly proteins and some fat for the stomach. And absorption is interesting because there is some absorption happens in the stomach. Alcohol absorbs actually quite rapidly in the stomach, and so is the aspirin and like ibuprofen, non-steroid anti-inflammatory drugs. And but as you can see, majority absorption is here, but so still to a certain degree. And then the motility will be that mixing and propulsion into small intestine. So here's we are in the small intestine, a lot of different enzymes. We'll talk about them a little bit later, and also you guys going to talk about that during your extra activity. Which one did you pick? For the extra curriculum activity, extra point activity, which two did you pick?
I don't know exactly what it's named.
Okay, all right.
Sorry.
All right, no worries. So digestion, so this is where the peptides, carbohydrates, fats, and nucleic acids get digested. So as you can see, stomach is a pretty heavy lifter here, digestive system. Absorption, peptides, fatty acids. And also notice here, no absorption and no digestion of the carbs here. Amylase does not like the acidic environment. So it kind of skips. It starts here and then continues here. So the carbohydrate digestion is kind of not happening in the stomach, waiting to be dumped into small intestine and for the pH to get neutralized, and then it can get picked up digestion again. So here we are, and of course the motility. And then in large intestine, mostly it's the mucus production by goblet cells. Not the human-made digestion, but yes, bacteria, right? A lot of digestion by bacteria, and we absorb molecules made by bacteria, mineral water balance, that usually happens in the large intestine. And of course, motility. Please be sure that you have this slide. Yes?
For oral cavity?
Yes.
What did you say about indigestion?
So indigestion, right, carbohydrates as it's here, comma, minimal fat, because of the lipase presence. Lipase is the digestive enzyme for fat, and absorption also, right, minimal, not none. None is not correct. Mm-hmm. Okay. So as we just talked, chemical and mechanical digestion begins in the mouth, right? And the saliva is very important, but it is under autonomic control, meaning autonomous nervous system. So it softens lubricate food. It will start digestion of the carbs, thus amylase, fats, thus the lipase. It also have a protective function because it has a lot of chemical that has actually bacteriocytic activity. And that's why if you see some people like licking their wounds, then it's okay. That's what they're trying to do. and of course chewing and swallow deglutition. The saliva production, as you know, under autonomic nervous system control. And with autonomic nervous system, the presence, the food, the smell of the food, it will activate the parasympathetic nervous system. So in the stomach, storage about 2-4 hours. Digestion, of course, lipid and proteins, not carbs. Amino acids, amino acids, of course, protein and amino acids. And then production of different enzymes like pepsinogen, pepsin, hydrochloric acid. There are also some hormones that we're going to mention a little bit later, and so you're also going to mention on your extra point activities, so I'm going to not focus on it right now. And also pH, also very important in defense against the swallowed pathogens, because it could neutralize quite a few of them just by hydrochloric acid. Okay, let's look at the question. Raise your hand if you want to answer, and let me see my little.
So, Professor.
Hold on a second. Okay, Ben? Oh, I'll get to. I think I have more questions. Ben?
C.
Okay, let me read. Secretion refers to release of substances into the lumen of the gut. We're okay with that? Okay. Angel? Yes, large intestine. Okay. So we are in a gastric mucosa here. Here's our gastric gland, we call it. And the reason we call it gland because it has a lot of cells that's going to secrete something into this opening. So the very first layer that is facing lumen would be mucus cells, right? So there's, key substance that they will be releasing is mucus. And that's super important for creating this barrier between the acidic environment of the lumen and the endothelial cells. When the mucus cells are not producing enough mucus, we could have erosions due to the acid presence, right? So very important physical barriers. And it's a tonic release, it makes it all the time. So then towards a little bit the deeper side of the gastric gland, we have a different cells, we call those mucus neck cells, and they actually make bicarbonate. So that is the sort of antidote to the acidic environment. So mucus and bicarbonate, so they are these two protective layers to protect the endothelial cells from, I guess it could be called epithelial cells from the gastric acids. Now finally, this shown here on the brown here is the parietal cells. Parietal cells make gastric acid, very important to actually produce pepsin, because as you can see here, here's pepsinogen. In order for that to get to a pepsin, you need to have the hydrochloric acid here. So, and then there is also an additional intrinsic factor that's made by parietal cells that is important for the B12 absorption. Enterochromaffin-like cells, and much less numerous in the gastric glands, they make histamine that actually stimulates the gastric acid. So it's all, as you can see, kind of intertwined, right? And then finally, Chief cells, I'm thinking like the chef. Chief cells would then make pepsinogen that will be converted with by the hydrochloric acid into the pepsin, and that in turn will digest proteins, and they also mix the gastric lipase to digest fats. The the D cells, right here, kind of little more elongated cells, not very numerous again. They make the substance called somatostatins, and they actually will inhibit gastric acid secretion, make sure it doesn't go crazy. And then the gastrin by the G cells, gastrin, G to G. So they usually kind of on the bottom of the gland. They also stimulate gastric secretion. So you have cells that stimulate and inhibit gastric acid secretion. So it's going to be all this interbalance between the cells. Now there is way more different substances that is made in the gastric mucosa and GI. I mean the small intestine mucosa, and this was just to give you a glimpse. But if you looked at the extra point activity, I've listed multiple, this and more different compounds that made by either stomach or the intestine. So I'm not going to focus on that because that's, guys, going to be your job for 20 points if you wish to do so.
Professor, now I know which one I chose. Is the cholecystokine, CCK?
Yeah,
and
CCK
and the GLP-1.
and GLP-1.
Yeah.
All right.
I was just measured for the
The Ozempic. The Ozempic. The Ozempic. All right.
Hard.
Ryan? Chief cell secrete pepsinogen. What do you guys think? Good.
Professor,
Yes.
you said that pepsin is converted with HCl to make what?
Pepsin. So see, pepsinogen, it's a precursor of pepsin. Like, see the way it's even
Oh,
written,
yeah,
they
yeah.
kind of.
So it's
So-
converted with HCl. to
Yeah,
make
yeah,
pepsin.
it's a precursor.
Gotcha.
A small modification, and off you go. Viv? G for gastrin, yes. Good job. All right. So this is the parietal cells, right? And what do you guys remember about parietal cell? Makes hydrochloric acid. It makes most stuff too, but hydrochloric acid is what we're gonna talk about. It's a pretty, actually simple mechanism, because the first thing is to get sodium. So first of all, kind of to orient ourselves. So this is a capillary ECF parietal cell lumen. That's a lumen of GI. So for the hydrochloric acid production, we actually have a relatively simple mechanism here. So we have this antiport, the hydrogen exchange for the potassium. And why do you think the energy is used? Antonella, what do you think?
I mean, I'm just gonna say, like, maybe like for the sodium potassium pump.
This is not a sodium
Active
potassium pump, but why
transfer?
do you think energy is
Active
used?
transfer.
Active transfer, but why do you need an active transfer? Why can't you just let diffusion take
Is
place?
it going
Uh-huh.
against its concentration?
Against the concentration gradient, because potassium goes against the concentration gradient. So anyway, once the hydrogen is in here, then there is another antiport that will deliver the chloride. Again, as always, you don't really need to memorize this transporters, just to know that so the hydrogen and the chloride will then make a hydrochloric acid. It specifically happens in the parietal cells, the very rich and the CA enzymes to convert the water into the bicarbonate, because that's the exchange for the chloride. So the other things that I'm pointing here, during the active digestion where we have a high concentration of hydrochloric acid is produced, we see something that we call alkaline tide. That's when there's a lot of bicarbonate exchange for the chloride, and the bicarbonate makes its way to the capillary. So if you measure the pH of the capillaries near the stomach, that's what we call alkaline tide. It's very alkaline. That's because of the movement of bicarbonate here. So, so quite simple mechanism of the making hydrochloric acid. I think it's the same. I'm not sure why I picked this one. I guess I just want to point out, remind you where the parietal cells are. So this is to remind you about this mucus bicarbonate barrier, super important. So here you have the gastric juice, very acidic, and this is bicarbonate, mucus bicarbonate barrier. So you have the mucus produced and the bicarbonate. And so that is the protection layer from the digestion of the, so that you don't auto-digest your epithelial cells. So at the surface of the epithelial cells, pH is quite normal, 7.2, 7.3. Normal for the body, of course. So do you guys remember when we were looking at the pancreatic islets, we said, oh, the glucagon and the insulin are made here, that's all good. But then we also mentioned that these are called exocrine glands, right? So these exocrine glands produce a lot of different digestive enzymes, a slide to follow, and they will then send them into the lumen of the pancreatic duct. And then pancreatic duct will drain into duodenum to do a few things. First, to actually secrete bicarbonate to neutralize the pH of the chyme, and second, release a lot of different digestive enzymes such as trypsin. So here's another example of the pancreatic duct cell. The cells of duodenum also can secrete bicarbonate as well, but so is the pancreatic cells. So bicarbonate is exchanged for the chloride. If you guys recall, when we talked about the cystic fibrosis, we talked about how the sodium potassium chloride co-transporters were actually, where is it here? Sodium, potassium, chloride. So that's why there's two C's here. This this transporter is actually mutated. Wait, what was I gonna tell you? I kind of lost my train of thought. Why did I jump to? Hold on, let me think about it. Okay, so hopefully come back to me. So this is a pancreatic cell. So the production of bicarbonate comes into the lumen and exchanges the chloride. As I mentioned, changes the pH, and the hydrogen is excreted. Oh, I guess it's not on the slide. I was like thinking, oh no, it is. I was like, well, it should be here. Because the hydrogen is actually excreted and it gets into circulation, circulation. So this is a very important part of neutralizing of the chyme. What was I thinking about? Let me think for a second. Oh, okay, come back to me now. Okay, so remember how the cystic fibrosis, what we talked about during the mucus production in the lung? A similar situation actually happens here in the digestive system and the people with the cystic fibrosis. So this particular transporter that exchanges the chloride, that moves the chloride into the system. So because they have a low production of the hydrochloric acid, there is a potential for more digestive infections, and that's another complication. So the sticky mucus and respiratory infections, and not enough hydrochloric acid and the digestive infections as well. So that's the second kind of main a reason for, like, main symptom for the cystic fibrosis people, and that's because of this chloride channel is mutated. Okay, so this is the example of sodium chloride secretion, and the sodium chloride secretion, and the sodium chloride secretion is kind of more like a lubricant, and in this case, here's a lumen. Sodium actually goes paracellularly, so between the cells, attracts the water with it. Chloride goes through the same channel like we just talked about. And so this sodium chloride is, we call it very often just like saline. It's very important for lubrication and kind of making sure everything's working in the moist environment. Okay, let's keep applying your knowledge. Stephanie?
Is it A?
In the digestive system, large amount of hydrochloride is released by parietal cells where there is a secretion, large amount from pancreas. So what do you guys think? Yes. See. Good job. Okay. Another secretion, bile. So bile is made by hepatocytes, but it is released through the gallbladder, obviously. So it's collected in the gallbladder and released through the bile duct. Why am I not talking today very well? Okay. So produced by hepatocytes, very crucial role as emulsifier to break down the big lipid mixtures into smaller molecules. To the degree, it also helps to neutralize hydrochloric acid as it enters to a duodenum. Here's the stomach ending, and this is a duodenum, right? So potentially could develop the gallbladder stone, of course, precipitate bilirubin. That's where the color from is kind of very dark, brown greenish color. So we already mentioned saliva amylases. We mentioned the stomach chief cells. This is all part of the secretion. Pepsin. These are the multiple enzymes that actually released by the exocrine cells of the pancreatic gland. So in addition to bicarbonate, pancreatic amylase, which is different from the salivary amylase, but it does the same thing. It digests the carbs. Trypsin, very important to digest proteins, lipases for lipids, maltase for maltose, lactase for lactose, sucrase for sucrose. So this is the carb, right? And this is its respective enzyme, a different ending. And nucleases to digest nucleic acid. So where do we get nucleic acid from, guys? Where? But where? Like, where are they coming from? How do we get them?
Nitrogen.
How do we get nucleic acid? Why do we need to digest them? Huh? Cholesterol? Mm-mm. Nucleic acids are DNA and RNA, correct. But how do we get them? Food, more specifically?
Protein.
Not protein.
Amino acids.
You're very close. Food,
Protein.
because we eat the DNA of the
Meat.
plants and animals, right? So they have the cells, and inside of the cells there's a DNA and RNA, right? So that's where we get them from, right? We get them from, I mean, food is correct answer, I was just trying to get a little closer. So because hopefully you understand, the DNA and RNA are mostly in the nucleus, of course, some of them are mitochondria, but as we eat other DNA and RNA containing animals and plants, that's where we get it from. We get it from, we get their DNA. Okay. This is a important little modification of the circulatory system that helps us to make sure that as we absorb all that stuff that we absorb, it goes through a little bit of a checkpoint, being a liver. So check this out. So here's our aorta, right? So sends all this arteries in different direction, and the hepatic artery does supply the liver. But notice this: as the arteries go to digestive tract, there is a, what we call hepatic portal system that goes directly, the venous blood picks up all the nutrients from here and goes into the liver before it gets to hepatic vein and before it gets to circulation. So as you can see how this is actually sort of like the checkpoint. The liver is, of course, the major organ for metabolizing a lot of different compounds that get through our ingestion. So let's see if there's anything. All blood leaving the intestine and stomach collected in a large vein that fused to form hepatic portal vein. So I hope you guys appreciated that. That's like all the veins from the digestive system goes through the liver first for the metabolism and detoxification before it can actually go back to the hepatic vein and an inferior vena cava and then hit the circulation. Very important setup. Let me see how we do it. Okay. Maybe we can do a few more slides. Okay, so this is one single kind of sort of the unit that we can look at the liver, right? So these cells that kind of yellowish browns, they call hepatocytes. They are main functional cells of the liver. And as you can see, the cells, they face this enlarged venous, special venous vessels, what we call sinusoid, because they're a little bit larger than usual veins. So then the hepatocytes have an opportunity to interact with these sinusoids, right? And here's your hepatic portal vein, comes in from the digestive tract, branches into all the sinusoid, and the hepatocytes have an opportunity to interact with every hepatocytes had an opportunity to interact with these compounds that brought from circulation. So this is how it goes, right? Absorbed from gastrointestinal tract. All the nutrients, bilirubin, drugs, foreign substances, anything that is absorbed through the portal system goes into liver. So some nutrients, of course, are going to be used to, let's say glucose will be built into glycogen for storage, right? Fat metabolism, make a protein, so amino acids gonna come in, and liver makes a lot of proteins. Do you guys recall that? A lot of different proteins, transferrin, albumins, immunoglobulins. Any hormone synthesis that is done in the liver could be using all these nutrients that you were just ingested. Urea production, detoxification, storage, especially for the glucose and fats. So then once this is done, it can go to a peripheral tissue. So of course, a certain amount of glucose will be released, the plasma proteins that just made in the liver, albumin, clotting factor, angiotensinogen, all that good stuff that was just made in the liver from the nutrients that you absorbed is going to make to the peripheral tissue, right? Vitamins, different metabolites. And hepatic artery, which is, if you guys recall, the branch of the aorta, will bring also,
I said celiac trunk.
good job. It will bring different metabolites, hormones, drug nutrients, because this is the central circulation, right? And then, if something should not go to hepatic vein and into the circulation to peripheral tissue, it will be excreted through the bile. So the way to excrete, of course, is through the bile. Okay, let's look at the question. Tyra?
A.
What do you guys think? Michelle? B? Brianda? A? Okay, what do you guys think? A.
Wait, why is it A, Professor? Isn't it like, I thought it's made in the,
It's not made in Galbara.
Oh.
It's only stored there. It's only stored there and will be released when the time arrives. It's just a place to kind of hold it. Okay, all right, so something that's a bit more easy to understand. So here you are just ingested carbs, right? Starch, glycogen from the animals or plants you eat, cellulose, you know that non-digestible fiber from, like, if you remember, like broccoli, and then when you kind of pull this thing, right? Like this ribbon-like structure, or especially in celery, that's the cellulose, right? Table sugar, of course, lactose, maltose, fructose, carbs. So you just ingested them. They have to get to be monosaccharides, meaning, like if this is, let's say, lactose, you just had a milk, by the action of enzyme lactase, it has to be broken to glucose and galactose. So monosaccharides meaning only one, like a simple sugar, monosaccharide. This is a building block, right? So these are the disaccharides. They're very easy to digest because they only have two simple sugars connected. So the sucrose, the table sugar, is the glucose and the fructose. So sucrase will do the job. Maltose, under direction of maltase, will be broken down to two glucoses. And then starch and the glycogen, amylase, will break it down to the glucose. So all carbohydrates will have to be absorbed either as a glucose, fructose, or galactose. So this is all simple sugars, also called monosaccharides. The cellulose, the non-digestive fiber, roughage, as some people call it, right? Doesn't get really absorbed in the small intestine, because it's it helps the chyme to move. It really helps to prevent the blockage, and kind of makes sure that the chyme moving nicely. But in the large intestine, it is digested by bacteria, and the byproducts are absorbed. But not, we don't digest them as humans. Okay. So then how do we absorb them? Here's our lumen in blue, ECF, and the capillary. The fructose enters on the special transporters. It's called the GLUT5. That's the one that kind of enters, and then it exits on yet another transporter, GLUT2. The galactose or glucose, they do have the importer with sodium. We saw that early on somewhere. I think when we talked just first about the transporters in general, that was one of the examples. And sodium potassium pump will normalize it, and the GLUT2 on the other end will move glucose into the circulation. So all carbs have to be digested to glucose, fructose, and galactose, and they move as a monosaccharide using dedicated transporters. They cannot get via diffusion. Yes? Bayman? Amylases, the enzyme used to digest are secreted by salivary gland, gastric glands, pancreatic and intestinal cells and muscle. Which one you choosing? D? D as in dog?
He said E.
E. E? I don't think that's what you said. Can you say it loud one more?
Yes, E, the salivary gland into the mouse and pancreas into the intestine.
Okay. Salivary gland in the mouse, pancreatic intestine, correct. So, proteins, we ingested the proteins, what do we have to do with it in order to absorb? See, we have to digest them. What do we have to digest them to? We actually have a few ways to deal with it. So, once they go through the hydrochloric acid, they will change their 3D tertiary structure and they kind of unfold. And here we have a few opportunities. Di and tripeptides, where are they here? Okay, so the di and tripeptides would be just the 2 or 3 amino acids.
In the cell.
Uh-huh.
On the bottom right.
This was right?
On the cell. Back what's written, there's a couple of them floating down below. It's like in the white box, and where the cell picture is.
Yeah, here, yes, but I was just trying to find it.
Think of it
But
on the bottom.
just look at this. Here's the typical peptide, right? So it will have a hydroxide group and a nitrogenous, and so the enzymes they're able to cleave peptide bonds, right? So this would be example of tripeptide. This is, we're not gonna call it tripeptide because it has only 4 amino acids. So that would be like just the small proteins. Excuse me, sole peptides. Peptides is actually like a shorter version. When we use the term protein, we usually refer to the much more complicated tertiary structure, or tertiary or quaternary structure. So once we have a protein, if we have the di or tripeptide, there is actually cotransporters with the hydrogen, so they could be moved in the di and tripeptide formation. Amino acid, the singular amino acids, they can also be co-transported with the sodium. Of course, sodium potassium pump in the background to normalize that. And if there are small peptides, so 4 or more, then they could actually be carried across by transcytosis, right? Forming the digestive vesicles and kind of opening up on the other side. And they can get to the circulation that way. So proteins do not have to be all digested to a single amino acids. So dipeptide, tripeptides, small peptides, or amino acids, all can be digested, but in a different format. Some via transporters, some via exoendocytosis combination, which we call transcytosis.
And that was a 4 or more peptide?
4 more. Yeah, because then the di and tri can go through the transporters. I'm thinking we're going to not do lipids today because they're a bit tricky here. But we have just I think lipids to finish this portion. Let me just check really quick. And we do have two lecture periods for the 22. I think we'll be okay. Okay, guys, please start reading, and then see you on Thursday or in the lab. The lab is wet lab, okay? Make sure you have your PPE.